for 4 years. He has served as president of the National Academy of Sciences for 12 years, where he was instrumental in developing the landmark national science education standards that have been implemented in school systems nationwide. Just this week, he was awarded the National Medal of Science, the highest U.S. honor for scientific achievement. He serves on the advisory boards of more than 25 nonprofit institutions, including the Gordon and Betty Moore Foundation. He is now the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California, San Francisco. Everyone, please give a warm welcome to Dr. Bruce Alberts.
Thank you. Very good. Thank you. It's a pleasure to be here. Can you hear me with this mic? Okay, great. I'm going to give you a little bit of personal history because young people like to know how people like myself got into their career, and especially like to hear about all the mistakes that we made because you're going to make mistakes too, so there'll be a little bit of that too. I was a high school student when this famous picture was taken of Watson and Crick. Of course, they solved a major mystery in our understanding of life, how the genetic information was stored in a DNA double helix. I'd like to point out that Watson at this time was only 25 years old. I'm a big believer in young scientists. We'll talk about that more. So they solved the mystery theoretically, but they didn't know anything about the machinery that made it possible to copy the DNA double helix from one generation of the cell to the next. And starting as a graduate student in 1961, I would spend more than 30 years working on exactly that problem. So I just want to emphasize how we thought about the cell when I started in graduate school and compare that through this talk with what the cell is really like quite differently. So we thought the cell was just a very concentrated mixture of proteins and nucleic acids and small molecules, all colliding with each other very rapidly, not organized. So the cell was full of biological molecules, just like a tiny test tube. And therefore, we thought how DNA replicated using the enzyme DNA polymerase, which was discovered shortly before I started my own work, is that one DNA polymerase molecule would randomly collide with the end of a growing primary strand and add one of the 4 nucleotides that would match the template strand. That was the magic of information transfer, that, of course, one strand of DNA, as predicted by Watson and Crick, would carry the information for making a new strand. But after one DNA polymerase molecule added in a nucleotide, it would immediately fall off, and another one would rapidly collide, and so on, all the way down the chain. That was how all life was supposed to work. So I had a very dismal career as a graduate student at Harvard because I was inspired by Watson and Crick to make my own theory about how it was possible for DNA polymerase, a single enzyme, which in a test tube would only work on single-stranded DNA, how would it be possible for it to replicate DNA inside the cell, which of course is a double helix, 2 strands, not base paired with each other. So I did many experiments trying to see if my theory was right. And of course, the many no answers did not surprise anyone, and it did not add to the store of scientific knowledge. Nevertheless, my professor, after 5 years, said, well, write a thesis, put on all the experiments you did, you're finished. And so I arranged to go postdoc in Geneva to work as a postdoctoral fellow and continue my work on DNA replication, I hoped. All I had to do was walk into a room with some famous professors at Harvard and have this little oral exam on my thesis, which no one had ever failed. So I had already packed up all my stuff. I had given up my apartment lease the next Saturday. I had bought my tickets to Geneva. I had a wife and a 1-year-old child. And to my great surprise, they didn't pass me. So I said I had to stay and do some more experiments. So my major point was, in looking back, and I wrote an article about this failure was a formative event in my career as a scientist because it made me think very hard about what I had done wrong and also about how I could do better next time. I spent about a month just writing things down and trying to figure out, could I really be a scientist? If I was going to be a scientist, what would I do differently next time? It's all described in this little article. So what I learned most of all is, of course, you can't predict biology. It's so complicated. Watson Crick, that was an unusual case. You have to discover by experiment how life works. It's so complicated. But most importantly, doing science is all about having a good strategy. That's the key to success. There are millions of experiments you could do. You should only do very few of those. It's like being a general in an army. The fun of science, a lot of it is designing your own strategy and seeing if you can make progress against this great unknown that's before us. I then finally, 6 months later, did more experiments, did get my PhD, did go to my postdoc. I was in Geneva for a year only. And almost as soon as I got there, I found that my hypothesis that DNA polymerase alone would copy DNA, the hypothesis on which I had based my thesis project, was wrong. Because geneticists had shown that DNA replication requires much more than DNA polymerase. And that had been done with this bacteriophage T4, a large double-stranded DNA virus that infects the bacterium E. coli. This was one of the founding organisms for the start of the field of molecular biology. And there had been this beautiful work done right in the middle of my thesis project, published in Cold Spring Harbor Symposium, that had shown that by genetics, many different proteins are needed to replicate DNA in addition to DNA polymerase. I hadn't read this paper because I had been reading biochemistry. I was working in a chemistry lab, and I didn't read genetics. So a second lesson besides having a good strategy is make sure you know what other people have done and build on it. Here was a major mystery I would spend much of my career working on. There were at least seven T4 genes that were absolutely required for replication of this T4 virus when it infects E. coli. Nobody had any idea what they did, so they gave them numbers, 32, 41, 43, and so on. Just before I got to Geneva, one of these, the gene 43 protein, had been shown to be the DNA polymerase made by that virus. And so the question is, why are there at least 6 additional proteins needed when this virus infects E. coli? So the obvious answer was DNA replication must be much more complicated than anybody had imagined. And I should say I wasn't alone in my belief that DNA polymerase was the only enzyme needed. That was the way we thought, very simply. Life was supposed to be simpler than it turned out to be. So after a one-year postdoc in Geneva, I started my own small laboratory as an assistant professor at Princeton University in 1966. I was 28 years old. This is a major message. We got to enable young people to do that again. The students and I focused on purifying each of those 7 proteins using the genetics as a guide. Our dream was that one day we'd have all 7 proteins purified, mix them together with double-stranded DNA, and we'd be able to get it to replicate. This is a picture of my lab at Princeton in 1969. Can anybody see me? Nobody could recognize who I am. I'm the guy in the red shirt with the white suspenders. The rest of these guys are undergraduates and graduate students. So the results of 15 years of biochemistry was to actually figure out what all these proteins did. There's a whole group of them that form a DNA polymerase holoenzyme, the polymerase plus a clamp. There's a protein that coats single-stranded DNA to make the template bases easier to replicate, and there's this activity called a primosome with a DNA helicase and a primase that is very important to enable double-stranded DNA to be replicated and opened. And so the end, we discovered that the DNA is made by what we call a protein machine. It's a complex of proteins that works just like a regular machine. Parts move relative to each other in an organized way using energy to drive them. two DNA polymerases work at the same time because the two strands run in opposite directions, as Watson and Crick had shown. So the one on the bottom in the slide could go continuously at 500 nucleotides per second along the leading strand. And the other one, because the DNA polymerase only goes one way, has to make the other strand, the lagging strand, backwards as a series of short pieces, which are later sealed together. But this whole machine is just like a little sewing machine. Everything stays together, so it's not the case that these DNA polymerase molecules add nucleotides and fly off. They both just stay there for a long period of time to make the reaction much more efficient. So all this is just an excuse for me to show you a movie, because this movie, which is my favorite movie, really illustrates how life works. It's not just about DNA replication. This is a picture of the DNA replication fork moving at the same speed as it does in each of your cells, and billions of your cells right now in your body, otherwise you wouldn't be alive. And this is the way that protein machines enable chemistry of life to be effective. So I could talk to you, and you could listen. So here it goes.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Well, you can see why that's my favorite movie. Partly because it was a complete mystery that we worked on for 30 years and years to try to figure out, and the answer, completely unpredictable, turned out to be quite elegant and beautiful. So we now know that the same mechanism is used to replicate DNA from large viruses like T4 to yeast to mammals. However, as more complex organisms evolved, each function in T4 was carried out by more proteins. So it turned out that T4 was a great shortcut. And I just want to emphasize to students who keep on reading these textbooks and taking exams that you can't forget the beauty of science. And that I think that movie is beautiful. This is a wonderful quote about the joy that comes from understanding through science the world. And this is from the Nobel Prize-winning physicist Richard Feynman. He said, the world looks so different after learning science. For example, trees are made of air primarily. When they are burned, they go back to air, and in the flaming heat is released the flaming heat of the sun, which was bound in to convert the air into tree. And in the ash is a small remnant of the part which did not come from air, that came from the solid earth instead. These things are beautiful things, and the content of science is wonderfully full of them. They are very inspiring, and they can be used to inspire others. I find that movie inspiring. I mean, it's a shame the vast majority of human beings never know anything about how the cells in their body work, because it's a beautiful story. So some personal lessons learned. First of all, go after mysteries. If you're going to do science, work on something that nobody understands. If you understand it, you'll make a contribution. Secondly, there are remarkable homologies between living things. Therefore, use model organisms wherever possible. I use a bacterial virus. Many people today are using yeast and Drosophila, fruit flies. Thirdly, nearly all cell processes will be based on elegant mechanisms too hard to predict. You just have to do experiments to decipher them. And we now know that almost every process in the cell is driven by a complex of 10 or more proteins. There are thousands of protein machines in a cell. These protein machines function very much like the machines in everyday life. They're driven by electric energy. They need to have directional motion to be effective. And their ordered movements come not from electricity, but by the energy of ATP or GTP hydrolysis, which is tightly bound to one or more of the proteins to make them move in one direction through allosteric shape changes. And so an important challenge for the next generation of cell biologists is to obtain the information needed to accurately describe the mechanism of every type of protein machine in a cell. We know a lot of the pieces. You could draw arrows. Here's a bunch of pieces that work together. They bind together and so on, but we don't know how they work. The only way to find out how is to reconstitute the many hundreds of protein machines from their purified components, as we did for this machine, so that you could figure out how they work in a test tube. So I want to talk a little bit about what I learned from writing a textbook. I've been doing this since 1978. And when you write a textbook or you give a course, and I'm a big believer in teaching, you learn a lot, and that's very important for your own research and your thinking, broadening your thinking. These are the first 4 editions. We spent huge amounts of time on this. This is the first set of authors crossing Abbey Road, because that's where much of the book was written, near Abbey Road in England, in London. And at the back is that famous Jim Watson, who's no longer 25 years old. It was his idea to write this book, and these are my fellow authors. One recent surprise for textbook authors of many is that all of the interior of a cell, just speaking broadly, is organized into many, many biochemical subcompartments. And this organization is largely done by what we call scaffolds, scaffold RNA molecules, scaffold proteins. And these compartments reorganize as a function of what the cell is doing. So they're located in different places, but they concentrate reagents to do specific reaction. Therefore, a cell is nothing like a test tube. It's quite more sophisticated. So just a second about scaffold proteins. This is a diagram from our textbook. The basic idea is that these flexible polymers, RNAs, or protein molecules bind other molecules, proteins and other molecules that are going to react with each other. Since they're bound, they're kept in close proximity, they're colliding much more rapidly than they would otherwise, and then the product is formed more rapidly. And moreover, it's formed in a specific place in the cell where the scaffold is located. A famous scaffold that we don't understand at all, which is critical to lots of disease and basic science, is the scaffolds that are underneath each side of a synapse. Underneath the synapse on both sides there's a unique biochemistry. Lots of special proteins held there by proteins that are scaffolds, protein and RNA molecules. And that makes our memory possible in ways we don't yet understand. Scaffolds are also involved in forming many different factories inside the cell. This is just a picture of a cell nucleus showing where transcription is going on in a cluster and where replication is going on. When those processes stop, these factories go away. When you damage a cell and you form DNA repair factories that form in response to need. There's factories for splicing RNA, for editing RNA, and so on. We don't even know all of them. And very recently, in the last few years, we've come to realize that the mechanisms include the formation of a new kind of structure called a reversible amyloid, a cross-beta structure that helps to form many of these aggregates. This is from the work of Steve McKnight's laboratory in Dallas. And this is a very recent Science magazine showing that understanding of basic mechanisms in the cells is going to be crucial for us intervening effectively in diseases. This is a perspective writing about 2 articles in the magazine in September that increase our understanding of what's happening in Lou Gehrig disease, ALS, where these nerve cells are degenerating. And basically, what's been shown is that the fundamental mechanism involves these disrupting the function of one of these factories that's processing RNA molecules. Something you couldn't even think about trying to explain if you didn't know the basic understanding of the formation of these little factories. So we have a huge amount to learn, and it's going to have a big impact on our ability to help with human health. Last Friday, our book went to the printer, so I'm a free man for 2 years. This is the 6th edition. Each time we write an edition, we are humbled by how much we still don't know. And in this edition, we put a new feature at the end of each chapter called What We Don't Know, because we found that students, since in textbooks you tend to write only about what you know, students get the misconception that, well, everything's been solved. But in fact, those of us who've been thinking deeply about this think we only understand 10% of what we need to understand about cells. So these are just an example of the kind of important, profound questions that young people have to go out and solve for us. Wonderful challenges. So my conclusion, it will probably take most of the century to gain a true understanding of how cells and organisms work. We need much more biochemistry in purified systems. We're also going to need, I didn't have any time to talk about this, new quantitative methods for analyzing, understanding the enormous complexity of life chemistry. We know that these enormous networks form in the cell, both cell signaling networks, actin filament networks, hundreds of proteins interacting with each other. Even when we know all the details of the parts, the human mind is not able to figure out what the net result is. We call these emergent properties, and we need new ways of computational analysis, mathematical analysis that takes account of the spatial geometry to try to figure out what the net result of all that is and help us come to a real understanding of the complexity of life's chemistry. And so many important mysteries remain, and there are great opportunities for new scientists and health professionals, bottom line. So unfortunately, our present system strongly discourages risk-taking. I keep on talking about the importance of enabling young people to jump into what I call the white spaces, areas of cell biology that are important. You realize when you write a cell biology, nobody's working on it with modern methods. And it's because of the training inertia. Everybody works on things that they've already worked on. So we've got to do more to help enable young people to be innovative, and older people. In addition, the National Institutes of Health has been overemphasizing short-term translational biomedical research, where we really need to understand mechanisms if we're really going to make progress in improving human health. And this is one of my favorite editorials when I was at Science. Had Huda Zogby, who's a great translational scientist. He's a MD who studies Rett syndrome and only became a researcher by trying to help these children. So she's a prototype of a great translational research, and she recognizes that she can't do her work without more basic knowledge about the cell. And she has this wonderful quote, because she's Lebanese and she speaks Arabic, so she could talk about translation. She said, "The challenge in translational medicine is that scientists are trying to translate a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and expertise." It's a very, very wonderful way of talking about this. And she's decrying the fact that today many highly qualified basic scientists feel compelled to jump on the translational medicine bandwagon. So just one example of a model organism, importance of Drosophila. I worked on the developmental biology chapter in this edition. We're still far from understanding how cells work together to form and maintain tissues. They do all these incredible things. And it's so sophisticated. Many examples show that first working on a mechanism in a fruit fly provides a shortcut to understanding humans. And thus, in this new edition, I counted the number of references in the development chapters to Drosophila. There were 50 of them, where Drosophila has laid the whole foundation for understanding of basic development of humans. This is 4 times more than the next most cited organism, which was the mouse. And we must do something to rebalance the age distribution of U.S. independent investigators in medicine. I told you that Watson was 25. I was 28 when I started. Look at what's happened here. This is a startling, depressing slide. In the 1980s, there were 2,623 principal investigators running their own laboratory funded by the National Institutes National Institutes of Health who were aged 36 and younger. Today there are 600. We lost 2,000 of these people. We got to do something to restore that. My colleague Harold Varmus, Shirley Tillman, and Mark Kirshner and I have been working on trying to think of what we can do and trying to focus on that very important issue. A final topic, what I have learned about science, very quickly, this is a broadest point of view. Science and technology are much more important for society, for nations, and for the world than most scientists think. This is what I learned being at the National Academy of Sciences for 12 years in Washington from 1993 to 2005, where it took me out to all around the world to see what science was doing for the world. This is my final issue of Science magazine when I finished as editor-in-chief. Based on a trip I took to India with news staff, we saw all these wonderful things that outstanding engineers and scientists in India were doing in their society to try to improve the livelihoods of their huge number of very poor people, mostly working in rural areas. And there's huge amounts of innovation going on that need to be spread around the world. But science is much more than just lifestyle benefits. Every nation also needs much more of the creativity, rationality, openness, and tolerance that are inherent to science. What Indian Prime Minister Nehru called a scientific temper. I only found out about this Nehru thing when I went to India the first time in 1993. So Nehru was a scientist. He was the first prime minister of the free India, and he wanted science for his country, which is why India's built all these IITs and has all this capability. But he talked more than that. He wanted the people to have scientific values because in a diverse democracy, what he called a scientific temper is essential for making that democracy work. And here's some of the values of science that are critical: honesty, generosity, a strong demand for evidence with openness to all ideas and opinions, irrespective of their source. And here's my favorite topic, what I'm going to work on for the next 2 years, reinventing science education and what science education should look like in school. The students should be doing science. Noisy classroom, the teacher is a coach. These are 12th graders in San Francisco. Learning how to look for evidence, be rational human beings. I don't think they're going to be, I don't want them to be scientists. Only a few will be scientists, but they need to be able to think logically, look for evidence so they won't be fooled by people trying to get their money or their vote. And this election cycle is all about fooling people. Advertising agencies are spending vast amounts of money to fool people, and if we had a more scientific temper in our population, that we'd have a better democracy. Now, here's my favorite biology class. University of Minnesota is now teaching all of their more than 1,000 Biology 1 students in flat classrooms. They built a building with 16 of these flat classrooms. And this is a no-lecture environment where the whole class period is solving problems. There are many other ways of doing this that are not so extreme, but this is the way we should be thinking about all of our science classes, getting students of whatever age to think like a scientist and solve problems and be logical people who look and demand evidence for statements. So my favorite quote, I'm just at the end here, is from a book called Science and Human Values, published by the physicist Jacob Bronowski. He, as a young man in the British Army, flew over Hiroshima and Nagasaki and was incredibly depressed by the destruction that atomic weapons had brought. He spent 10 years reading and thinking and writing this book, asking whether science was good or bad for the world. And of course, he concluded that, on balance, science was very good for the world. And here's his concluding statement. He said, "The Society of Scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. From these basic conditions, which form the prime values, there follows step by step a range of values: dissent, freedom of thought and speech, justice, honor, human dignity, and self-respect. Science has humanized our values. Men have asked for freedom, justice, and respect precisely as the scientific spirit has spread among them. So this is just a picture taken about 15 years ago in a Swiss McDonald's. There were 15 Academy presidents from around the world. I could only get a small number in any one picture. These are the Academy presidents of India, China, Japan, and Sweden. The message is that we share across the entire world. Scientists, even I've interacted with the presidents of the Iranian Academy, for example. We share a common set of values, a common set of assumptions. We're all unhappy about our government making short-range decisions, not based on what science knows. And because of our common culture, we could communicate easily across the world. It's a great opportunity we need to exploit even more than we do today. And this is my last slide, a wonderful quote from Louis Pasteur about the role that science plays in the world. Thank you.